Uh, Good morning, all. I'm audible?
Yes, sir.
Yes, sir. So kindly accept our apology due to some technical issues. So the things has been halted. So now somehow we have resumed it. So shall we start this sessions? Morning, all. I'm audible. So
Yes, sir.
okay. So our first presentation will be Anjali Singh. Anjali, can you please start with your presentations? Anjali, you are there? Ankuresh? Archana Chaudhary? Archana, can we start your presentation? Archana, am I audible? Archana, can you start your presentation, please?
Hello.
Yes.
Hello, sir. Myself, Kirti Verma. Can I start my presentation?
Yes, you can start. Yes, you can start.
Okay. Let me, sir, share my screen.
Anjali, after Kirti, you can start.
Please on.
Anjali, please unmute yourself. Anjali, your voice is not coming. Please unmute. Kirti, just 1 minute.
Wait, sir. Wait, sir.
Anjali, we cannot hear you. Your voice is not coming, Anjali. Anjali, else can you connect with your mobile? Anjali, can you connect with? Okay, while
Sir, is my slides is presentable or visible on the screen?
Kirti, just 1 second. Anjali, you just exit the meeting and rejoin so that it will work. You can exit and rejoin so that it will work. No one can hear you. Anjali, I can. Now you'll be able to share your screen.
Is it visible?
Yes, you can start.
Okay, thank you, sir. Good morning to all the respected dignitaries, juries, delegates, and all the participants. Today, in the series of poster presentation, I, Kirti Verma, PhD research scholar working under the supervision of Dr. Sudan Singh in CSIR CMAP, Central Institute of Medicinal and Aromatic Plants, Lucknow, presenting my poster on the topic entitled Effect on Yield and Land Use Productivity Due to Spatial Arrangement of Cereal Crops in Kalmic-based intercropping system. Background of the study is Kalmig herb is a syaphytic in nature, possesses versatile medicinal property due to which its requirement upsurge in the industry. The initial fast growing rate and deep root system of cereal crops offers a good scope for intercropping with small herbaceous crop, early maturing and shallow rooted crop. Agronomic practices like spatial arrangement is known to affect the crop environment, which influence the yield and yield components. Optimum crop density should be maintained for the attaining higher yield and effective utilization of resources. In the context of sustainable agricultural system, intercropping is the most promising and self-reliant, a more profitable, energy efficient and cost-effective agricultural system that always has been the keen attraction center for the farmers, researchers, and the policymakers worldwide. Therefore, our present studies were planned to evaluate the yield advantages and land use efficiency of kalmik under different spatial arrangements of cereal crops for enhancing land productivity, sustainability over the present monoculture system of crops. Methodology of the study: the field experiment was conducted at the CSIR CMAP Lucknow, Uttar Pradesh, India for the 2 consecutive years, that is 2020 to 2021 and 2021 to 2022 in the month of July to January. There are total 9 treatments comprising intercrops with kalmik at 3 spatial arrangements of cereal crops. That is 1:1 ratio, 1:0.75 ratio, and 1:0.50 ratio, and 3 sole crop treatments. The 9 treatments are treatment T1 kalmik plus maize at 1:1 ratio, treatment 2 kalmik plus maize at 1:0.75 ratio, treatment T3 kalmik plus maize at 1:0.50 ratio, treatment T4 kalmik plus permanent at 1:1, treatment 5 kalmik plus permanent at 1:0.75, treatment 6 kalmik plus permanent at 1:0.50, and treatments T7 kalmik sole crop, treatment T8 maize sole crop, and treatment T9 maize pearl millet sole crop. These were evaluated in a randomized block design. Growth observation, herbage yield, and economics were recorded and calculated at the time of harvesting from each treatment. The land equivalent ratio, area time equivalent ratio, and land use efficiency was the criterion for measuring the efficiencies of intercropping advantages using the resources of environment compared with monocropping system. Statistical analysis of recorded data was analyzed by using the technique described by Fans and Sukhmate. Now the results of the experiment. The intercropping system and spatial arrangement of cereal crops had a significant influence on plant height of kalmik as well as intercrop, which is depicted in the figure number 1. The highest plant height of kalmik was attained by treatment T4, that is 75 centimeter, followed by treatment T1 and T2. And the lowest was found in the sole kalmik, that is treatment T7, which is 59.33 centimeters. The data of the kalmik plus cereal crop intercropping system revealed that the highest leaf area index was attained at the highest crop density, irrespective of the others. That is depicted in the figure number 2. Therefore, among all the treatments, treatment T4 was found highest, that is 3.03 LAI, followed by treatment T5 and T1. Next is intercropping of kalmik plus cereal crop was with spatial arrangement recorded higher land use efficiencies with the treatment T6. Land equivalent ratio was recorded 2.09. Area time equivalent ratio was recorded 1.56, and land use efficiency was recorded 1.83, followed by treatment T5 and T3, which is depicted in the figure number 3. Data pertaining to the herbage yield was depicted in the figure number 4 of kalmik, revealed that the treatment T6 found higher among the all, and the herbage yield recorded was 38.30 quintal per hectare, which was at par with the treatment T3, 37.81 quintal per hectare, and the lowest was recorded in sole cropping system of kalmik. With respect to the economics, which is depicted in the figure number 5, the highest net return was also attained in treatment T6, followed by treatment T5 and T3, and the lowest was recorded in monocropping system. Now the conclusion of the study is Result obtained from the experiment showed a significant advantage from intercropping for exploiting the resources of the environment compared to the sole cropping, which attributed to the better economics and land use efficiencies. Kalmig has intercropped with spatial arrangement of cereal crops like pearl millet and maize at 1:2.50 spatial arrangement. That is, in treatment T6 and T3 are found better among all for earning higher productivity and more profitability for the farmers community and their monetary gain also. Acknowledgement. Authors are highly thankful for the AgriVision, which provided us the platform to present our e-post presentation, and the director of CSIR CMAP to provide all the necessary facilities for carrying out this study, and CSIR New Delhi for the financial support under the CSIR Aroma Mission project. Thank you. Hello.
Yes, yes.
Hello. Any questions from anyone? No, sir.
Any questions from anyone?
No, sir.
So shall we proceed for next?
Okay, so still I just escape my slides, or
Yes, you can no, skip your slide so that no, we ask for the next participant.
okay.
এটা বেরিয়ে গেল।
Hi Anjali. Yes, Anjali, you can share your screen. Anjali, it's still loading. I think it's not visible properly. Anjali. Anjali, can you share your screen? Anjali, please unmute yourself and share your screen.
Hello.
Yes, Anjali, you are audible now.
Okay, sir. Good morning to all of you. Myself, Anjali Singh, and I'm pursuing my PhD from CSIR CMAP Lucknow, and I'm going here to present my e-poster presentation on the assessment of the effect of different agronomic intervention on Withania somnifera dunal yield and quality. Sir, actually, my slide is not. Now come to the introduction point. Ashwagandha dunal is an important medicinal plant, which is also known as a root of this plant are being used in Ayurvedic and other medical systems.
Anjali, just 1 second. I think your screen is not visible to anyone. So can you please stop and share your screen again? Yes, visible now.
Okay.
Now you can click on resume slideshow.
Okay, sir.
Just click on resume slideshow, so it will start.
Sir, is I visible? Is my slide
Yes.
is visible?
Yes, it's visible now.
Okay, thank you, sir. Now I'm going to introduce about ashwagandha. Ashwagandha runal is an important medicine which is also known as the Indian ginseng and winter cherry. Root of this plant are being used in properties. The root of the plant are categorized as a rasayan which are reputed to promote health and longevity by the augmenting defense against disease, arresting the body in a debilitated condition, increasing the capability of individual to resist adverse environment factor, and creating a sense of mental well-being. In Ayurveda, it have been internally for fever and hemorrhoid, externally for the wounds. A number of agronomic manipulation and practice have been tested aimed to improving the yield and quality of fruit of ashwagandha. The production of medicinal plant is economical, then the secondary metabolite obtained from the plant are acceptable. The favorable yield can be obtained by manipulating the agronomic practice. Now come to the point of the methodology. The field experiment was conducted both consecutive year 2019 to 2021 in month of October and March at the experimental farm of CSIR CMAP Lucknow. The experimental site was classified as a semi-arid tropical zone with extremely hot summer and fairly cool winter. The soil of the experiment site was sandy loam in texture having moderate fertility with 8 pH. Weather condition, temperature, relative humidity, rainfall prevailed during the year experimental period. The combination of the treatment comprises two varieties of ashwagandha, Nimitli 118 and Nimitli 101, then and 3 stages of harvesting, that is pre-flowering stage, flowering stage, and maturity stage of the harvest. The experiment was laid out in a split plot design with 3 replicate in individual plot site. The size of the seeds of ashwagandha V1 and V2 were hand sown uniformly in the line. Now come to the result. Yield attribute of ashwagandha influenced by the cultivar, plant population, and stage of harvest is fresh leaf yield, dry leaf yield. V1 perform better in comparison of V2, which is Nimitli 101, and in plant population P2 perform where the plant population is 250,000 plant per hectare, and the harvesting stage which perform better is a seed maturity stage. Now come to the economic point. V1 as from the same above, V1 perform better, and the net return obtained from the P1, P2, and S3 give better net return in comparison of other treatment. Overall conclusion obtained from the result table that variety Nimitli 118 and population 2 lakh seed maturity harvesting stage treatment combination so best result in comparison of other treatment in terms of growth, yield, and quality of ashwagandha. Now I would like to thank Director CMAP and my supervisor Dr. Saudan Singh, and thank you to all delegates. Thank you, sir. Hello.
Hello.
Yes. Any questions from anyone? Participants, any questions? Please come up with questions. Okay. So shall we move for next slide?
Any question related to my slide? Thank you, sir.
Any questions? Thank you. Thank you. So Ankuresh is there. Okay. Archana. Archana is there. Okay. Chaudhary Ankit Kumar.
Yes, sir.
Okay, you can start.
Okay, sir. Sir, I am visible and audible.
Yes, you are visible. You start now.
Yes,
Good morning, everyone. Myself, Chaudhry Ankit Kumar Umedbhai. I am research scholar at the Department of Aquatic Biology with Nirmal Sir Gujarat University. Today I am presenting e-poster presentation on study the effect of bean bran extract on the growth of Spirulina maxima in indoor culture. This is my abstract. First of what is spirulina? Spirulina is a microscopic, multicellular and filamentous cyanobacterium. Spirulina, a blue-green alga known for its high nutritive value, exists in freshwater, brackish water and seawater habitat. It has been classified under the kingdom Monera, the single-cell organism. There are 35 species of this genus. Scientific classification, see the microscopic observation of spirulina. Domain bacteria, phylum Cyanobacteria, class Cyanophyceae, order Spirulinae, family Spirulinaceae, genus Spirulina. Spirulina, two main species, Spirulina platensis and Spirulina maxima. Methodology. Media preparation, carry out elaborate study with wheat bran extract on the growth of Spirulina maxima. After drying wheat bran, 10 gram of the powder wheat bran mixed with 100 ml tap water and steam on water bath for 30 minutes. After cooling, filter through filter paper till clear solution was obtained. Autoclave the extract and stored as a stock solution of 10% wheat bran. See the image of wheat bran and wheat bran extract. We conduct the study by giving different concentration of wheat bran extract in 200 ml modified medium. Concentration wheat bran extract concentration 0.2 ml, 1 ml, 2 ml, 3 ml, and 4 ml, 5 different concentration. And observe the growth and chlorophyll concentration. The effect of 5 different dose of wheat bran extract. Dry biomass was noted highest 0.164 gram at 200 ml per 3 ml concentration. At the concentration of 200 ml per 4 ml, chlorophyll A 12.2692 milligram per liter, chlorophyll B 3.9454 milligram per liter, and C 1.8909 milligram per liter were observed in the highest concentration. See the dry weight and chlorophyll ABC high, which is highest compared to control. And chlorophyll estimation, we use the dichromatic method. Conclusion, the study of goal of accelerating the development of Spirulina maxima was significantly achieved by adding inexpensive wheat bran extract to modify media. See the image of we provide blue light for Spirulina culture with respect to indoor culture. This image first they inoculate Spirulina culture after 8-day growth observation of Spirulina. Acknowledgement, our thanks are due to Department of Aquatic Biology at Nirmal South Gujarat University for providing laboratory facility and other help. I would like to thank the Society for Agriculture Research and Management, AgriVision Center University for organizing this conference, which gave me an opportunity to share my research on this platform. Thank you to all.
Any other questions? Any questions from anyone?
I have a question.
Yes.
Hello.
Yes, sir.
Whatever you present, it is all right. But the data, whatever you have shared in your screen in the second slide,
Okay.
is it you have gone through the statistical analysis?
In Excel.
No, that is Excel is all right, but there is no standard error bar. Without standard error bar, the analysis is not complete.
Okay.
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 Now you are in which year of PhD?
Third year.
Third year. You must have to keep the standard error bar. Okay.
Okay.
If you, because without standard error or without statistical analysis, without giving the p value and cb value, the experimental data is invalid. Okay.
Okay.
So for the next time, you must have to give the standard error. Okay.
Okay. Okay, sir. Thank you so much.
Okay. Okay. Thank you.
Sorry, sir.
Okay. Wish you all the best for the future.
Any other questions from anyone? Okay, shall we proceed for next presentation? So, Maniratnam. Okay. Pushpal Singh.
Yes, sir.
Kushal Singh.
Yes, sir. Yes, sir. Sir, my voice is audible.
Kushal, you can.
Okay, sir.
Yes. Yes, you can share your screen.
Yes, sir. Sir, I am voice visible and audible.
Yes, clearly visible.
Okay, sir. Sir, good morning to all delegates present on over there. Myself, Kusalpal Singh, pursuing my PhD from CSIR CMAP Lucknow. Here I am going to present my e-poster presentation on title, effect of date of harvesting and age of leaves on essential oil content, oil yield and oil quality in menthol mints. Come to the introduction. Menthol mints is an important essential oil bearing plant that belongs to family Lamiaceae essential oil extract through hydrodistillation method and used in flavor and pharmaceutical industry. This has already arrested the decline in the area under mint cultivation, which expected to restored to previous level. The area cultivation of menthol mint 2,50,000 to 3 lakh hectare in the next few years, leading to revival of Indian mint industry. This crop play a crucial role in enhancing the economics of grower. There are few constraints are being facing by farmers in commercial cultivation because low productivity due to untimely harvest of this crop. A number of leaves were fall at late harvesting that cause in low essential oil yield. These constraints are reducing the productivity and profitability. Therefore, the aim of the study to optimize the date of harvesting and age of leaves for enhancing productivity and resource use efficiency of menthol mint along with the object to enhancing the overall productivity of menthol mint with a significant reduction in cropping period. Come to the methodology. The field experiment was conducted both consecutive year to 2018 to 2020 Feb. at the experiment farm of CSIR's Central Institute of Medicine and Aromatic Plant, Lucknow. It is located at 26.5 degree north latitude and 80.5 degree east longitude, 120 meter above mean sea level. The soil of the experiment site was sandy loam with texture having moderate fertility with 7.3 pH, weather condition, temperature, relative humidity, and rainfall prevalent during the year experimental period. The experiment consists of 5 dates of harvesting, that is 30 April, 10 May, 20 May, 30 May, and 10 June, and 4 age of leaves, small size leaf, middle size, big size, yellow leaves. We are conduct and randomized block division with their replicates. Result: plant growth parameters, plant heights, plant spread, fresh weights and leaves weight per plants, stem weight per plants, fresh salvage yield, D3 best, other compare date, oil yield kg per hectare, D3 date, oil content and menthol content influenced by different date of harvesting and age of leaves. Oil content in best D3 dates. Come to the conclusion. It is conclude from the current study that maximum fresh herb yield and oil yield were recorded when the crop was harvested at 20 May as compared with other harvesting date. Moreover, the highest essential oil content, menthol content oil yield and menthol yield was recorded in old leaves. Corresponding maximum gross return net and PC ratio were recorded when crop was harvested at 20 May. However, it provides an opportunity to check the exact date harvesting along with size of leaves. Overall, the treatment combination of 23 of harvesting and old yield leaf enhancing the productivity and resource use efficiency. Acknowledgement. The author are proudly thankful to the directors CSIR CMAP and my supervisor Dr. Sadan Singh and provide the necessary facilities, conduct experiment trial with financial support from CSIR CMAP. The project gap 0484. Thank you, sir.
Okay. Thank you.
Thank you, sir.
Thank you for the nice presentation. And please come to your last slide. Please come to your conclusion part.
Yes, sir.
Here, one thing I suggest, you have mentioned the date, na 20th May. It is not better. Okay. It may be 20th May or it is 15th. Hello.
Yes, sir. Yes, sir.
No, not question. Just a suggestions for you.
Yes,
You
sir.
just not mention the date. Okay. 20
Okay,
May, 20 May, 15 May. You can write it as a page. Okay.
Okay.
First page or third page, whatever. Okay.
Okay, sir.
Then it's all right. It's all right. Okay.
Okay.
Thank
Sir.
you.
Thank you, sir. Thank you, sir.
Thank you. Thank you.
So you can skip your presentation now. So Muhammad Abdul Wasim, Pooja Urade. Pooja is there.
Sir, it's Archana Chaudhary.
Yes, Archana.
Sir,
Archana,
sorry for the delay, sir.
No, it's okay. It's okay. You can start now.
Okay, sir. Good afternoon, everyone. Myself, Archana Chaudhary. I am pursuing my PhD from CSR CMAP at Lucknow. And I am here for presenting a poster on the topic of farming of geranium as a persistent crop under natural climatic condition of Indo-Gangetic plains. And this is the introduction. Rose-scented geranium, that is a Pelargonium graveolens, is an annual or perennial herbaceous multi-harvest, high-valued aromatic and medicinal plant that belong to the family Geraniaceae. It grows in tropical, subtropical, temperate climatic condition of the world. Its cultivation is done for obtaining high-quality essential oil, which is rich in geraniol and citronol. Its essential oil is used in cosmetics, soap, food, pharma industry, and in aromatherapy. It was originated from South Africa and introduced in India in the 20th century by two French nationals. Sir, my voice is audible?
It is audible. It is audible. Proceed. Proceed.
Okay, sir. It was originated from South Africa and introduced in India in the 20th century by two French nationals. In India, geranium cultivation is practiced in different parts, but till date, India is not sufficient to meet its domestic demand. Hence, India imported its essential oil from China and Egypt. To fulfill its growing demand, it is necessary to increase geranium essential oil production by cultivating it is a perennial crop at larger scale, particularly in the Indo-Gangetic plains. Thus, there is obvious merit in exploring the possibilities of perennial-oriented geranium cultivation in Indo-Gangetic plains under the semi-protective shade. Keeping above in this mind, the present investigation has been planned to grow Pelargonium graveolens as a perennial crop under shade by taking multi-harvest in a year to evaluate the effect of multi-harvest on yield and quality of geranium quality as a perennial crop under semi-protective shade. This experiment comprised of 4 treatments. This experiment conducted at the semi-arid semi-tropical zone with hot summer and very cool winters. The soil of the experiment site was sandy loam in texture, having moderate fertility with 8.0 pH. Weather condition included temperature, relative humidity, rainfall during the experimental period were noted. And this experiment comprises the different treatment combinations as multiple harvesting and different date, type of set. And the following observations were collected from the fields: growth parameter, yield parameter, and quality parameters. And this is the graph. The first graph is showing the herbage yield, fresh herbage yield, fresh yield of leaf, fresh yield of stem, and LS ratio, in which the maximum fresh yield herb yield was recorded at the third harvest, which was followed by the fresh yield of leaves and fresh yield of stem. And the LS ratio, highest LS ratio was recorded at the ninth is harvesting stage. And the same second graph was the essential oil content and geranium yield and citronol yield. The highest essential oil content was recorded at the D7 at the seventh harvest stage, and the oil yield was recorded at the third harvesting stage. This is the conclusion. The conclusion obtained from the result table concluded that the third harvest produced the maximum fresh herbage yield, that is a 14.04 ton per hectare recorded at third harvest, and the seventh harvest recorded the maximum oil content, and the first harvest had the maximum oil yield, that is 23.75 kg per hectare. The author is thankful to Director CSR Center Institute of Medicine and Aromatic Plant and AgriVision 2023, Century University and SARM for providing me this opportunity to present my e-poster presentation. Thank you, sir. So is my voice audible?
Yes, you are audible. Nice presentation. Okay. Just I want one query. Just let me know what is the difference between oil content and maximum oil content and maximum oil yield.
Sir, maximum oil content in which we taken is the sample of 100 gram sample, in which the maximum oil content are obtained, in which the small destination unit, but the oil yield was recorded from the hectare area, hectare area.
Okay, then what is the data you have mentioned here in the last slide that in the maximum oil yield is 23.75 kg per hectare, and what is the data of maximum oil content?
Maximum oil content, sir, 1 minute. So 0.19% that recorded as the
0.19%. 0.19%. How you have got this?
Sir, we actually taken calculation by 100 gram sample. We take 100 gram sample in a cleavanger apparatus.
Yes.
By distillation, we taken this sample by calculating the reading. First fresh herbage.
You have to mention this thing in your slide also. Okay. Now you have mentioned two data, that is the maximum fresh yield, that is 14.04 ton per hectare, and another is maximum oil yield, And according to the in addition to these two things, you must have to present in the slide that maximum oil content. Okay. And
Okay, sir.
one more thing, whatever the maximum oil content you get here, okay,
Okay, sir.
you have compared this thing with previous literature.
Yes, sir.
Acha. Okay. All right. Okay. Thank you.
Thank you, sir.
Thank you. Thank you for your presentation.
Thank
Uh,
you, sir.
So next, Moniratnam. It's not there. Abdul Wasim. Pooja Orade. Vartika Tripathi. Vartika Tripathi. Manasi Mandal.
Yes, sir.
Manasi.
Am I audible? Am I audible?
Yes, yes, yes.
Yes, you can proceed.
Okay. Can you visible my screen?
Yes, yes, but visible. You proceed.
Okay. Good afternoon, everyone. I am Manasi Mandal, PhD research scholar. I am pursuing my research under the guidance of Dr. Narottam Dey in Vishwabharati Shantiniketan. Today, I am presenting a poster entitled with Combined Effect of Submergence and Salinity Trace on Growth in Correlation to Carbohydrate Reserve in Rice Genotype. This is the abstract of my poster. First, introduction. All of we know that rice is one of the most principal staple crop in the world, especially in the Southeastern Asia. So in lowland coastal saline belt, plant faces either salinity trace or submergence trace individually or both. When plant are face submergence, in submerged condition, oxygen concentration in water is lower down or absent, so respiration of plant is inhibited. On the other hand, salinity affects on plant growth. Also reported that salinity also affect on oxygen solubility in water. So in combined stress is more detrimental in terms of plant germination, reduction of plant biomass, chlorophyll content, and photochemistry also. So the changes in fermentative enzyme and carbohydrate catabolism is important factor to tolerance the anoxia. So initially, the vast fermentative enzyme provide the higher ATP required as cell to activate the anoxia, but then the lower rate of fermentation would conserve the carbohydrate for long-term survival. We initiate this study with the hypothesis that the tolerance of ion toxicity and anoxia are independent and contribute equally towards tolerance to submergence by saline water flooding. For this study, I used 5 different types of rice seedling, namely FR13, a well-known submergence tolerant rice lines, which I give the accession number VV69, and Nona Bogra, a well-known salt tolerant rice line, I give the accession number VV170, and both the susceptible rice lines IR64 with accession number VV185, and the two landresses Kagabad and Dudeshwar I used for this experiment. First, I used 28 days seedling of 28 days seedling of all varieties, and after that, plants are subjected to soil trace, which having electrolytic conductivity is 12 decisiemens per meter, or the molar concentration of this salinity is 103 millimolar. And another set of pods are submerged with normal water, and another set again another set of pods are submerged with saline water having electrolytic conductivity 12 decisiemens per meter, or 103 millimolar concentration of water. For this for 7 and 14 days in respect to control. I give the trace for 14 and all these conditions for 7 and 14 days in respect to control. And after the stress, I study these physiological parameters like shoot length and internal length. And for the biochemical assay, I study the soluble and insoluble sugar with the standard protocol of EM and William, and fermentation and also I study the fermentative enzyme pyruvate decarboxylase and alcohol dehydrogenase was measured according to the method of Fukao et al. And the quantity of protein was estimated by the method of Hicks and Quiffet. So the result and discussion. There are the 4 color represent the 4 different phase of stress condition. There blue color represent the control condition, and red color represent the salt stress condition, and yellow gray color represent the submerged with normal water, and yellow color represent the submerged with saline water. That is the combined trace, and this yellow color represent the combined trace. Result shows that plant growth is more diminished in combined stress in both respect to 14 and 7-day stress induction in compared to the individual stress. Submerged tolerance rice lines FR13 that is represented here in VV69, and land races cover shows more so more growth reduction in combined stress in compared to the other variety. Plant increasing their shoot length by increasing their internode length. So in 7 days of stress induction, fast internode length of all varieties are more or less increased, but the susceptible varieties show more elongation of internode length. By elongating their internode length, they can increase their plant height. Same scenario seen in 14 days, but exception occur in variety of Nona Bokra that show second internode elongation, rather they show fast internode elongation in 14 days stress induction. But in 7 days in stress induction, fast internode of Nona Bokra is increased, and by this increasing the internode, plant can increase their shoot height. In the biochemical content, during the anoxia, no or less photosynthesis occur and respiration is inhibited. So plant use their conserved carbohydrate for their growth in terms of their elongating shoot length or by means of elongating internode length. Sir, am I audible? Sir? Hello, sir?
Yes, you audible. You proceed.
Okay, sir. Tolerant cultivar accumulate more soluble and insoluble sugar by reducing their growth compared to the susceptible cultivar. So total soluble and insoluble sugar shows significant positive association with survival percentage. On the other hand, elongation due to anoxia was significantly negative association with survival. Here we can see that the submergence tolerance rice cultivar FR13, that is the VP69, shows greater accumulation of soluble sugar and insoluble. That is the insoluble sugar in combined stress condition in compared to the submerged stress condition. But when plants are subjected to 14 days stress induction or more retention or with increasing the period of stress condition, plant reduce their soluble sugar. There is a reason because plant use their carbohydrate through the fermentative pathway because there is a lack of oxygen or deficiency in oxygen. So respiration is inhibited. So plant use alternate pathway by which the carbohydrate is broken down. And for this carbohydrate broken down, PDC activity is required. If PDC activity is more, then more carbohydrate is broken down. So tolerant variety shows less PDC activity, so they can retain or restore their sugar content which have been used for their future or long-term uses after when trace are removed for prolonged period of stress. So the tolerant rice variety shows the lower PDC activity in in combined stress condition in both the 7 and 14 day stress induction, but the rice lines or intolerant rice lines shows the greater activity of PDC by the greater activity of PDC, greater soluble sugar are used by the plant. in the other hand, by the activity of PDC, a toxic intermediate acetaldehyde is accumulated in the plant cell. But this toxicity is removed by the activity of ADH, which ADH means alcohol dehydrogenase, which remove the acetaldehyde by the converting the acetaldehyde to ethanol, which is soluble in water. So tolerant varieties show higher ADH activity to remove the toxicity of intermediate acetaldehyde. Here we can see the 7-day stress induction, more or less all varieties shows more or less same activity of ADH, means in combined condition ADH activity is greater in compared to the submergent stress, but prolonged period of stress induction, only the submergence tolerance rise lines FR13 show the higher activity of ADH in compared to the all variety. Here also you can see the in combined case, combined stress condition, ADH activity is more than the individual submerged condition. So from this study, we can conclude that in combined condition, elongation of plant heights was more diminished, and susceptible rise line shows more intermediate elongation than the tolerant line. And during combined stress, greater activity of ADH along with the lower activity of PDC facilitate greater accumulation of carbohydrate in tolerant rice cultivar FR13 by reducing of growth. So I can acknowledge a very humble thanks and regards to my supervisor for their excellent guidance in my research, and the funding agency of DBT and UGC, and organizing committee of AgriVision for giving opportunity to this excellent e-poster, to the presenting the e-poster. Thank you all.
Okay, thank you for the nice presentation. It is audible, Manasi?
Yes, sir. Yes, sir.
Just let me know what are the susceptible variety you have taken and what are the tolerant variety you have taken during your analysis.
Susceptible variety, that is the VB185, means IR64. This is susceptible for IR64. I have used a susceptible rise line IR64, that is susceptible to both submergence as well as salinity stress. And I use the tolerant rise lines, means positive check for submergence tolerance, that is the FR13. And for salt tolerant rise line, I used Nona-Bokra, well-known salt tolerant rise line. Nona-Bokra I used for the positive check for salinity trace. That is the VB170.
Okay. Why sub-1 you know?
Yes, sir. I sub-1.
Sub-1. Why you are not taking sub-1?
Sir, for my future study, I use the sub-1 locus in this variety, but I could not take any variety that have the sub-1 individually sub-1, because already FR13, a well-known rise submergence tolerant rise line, which have the sub-1 locus. So I take the FR13 for their check.
Okay. Okay. Thank you. All right.
Thank you. Thank you, sir.
Hello. Any further questions from anyone? No. Okay. So shall we have next participants? Bartika Tripathi. Okay. Pooja Urade. Muhammad Abdul Wasim. Maniratnam K. Katari Venubabu. Archana. I think Archana presentation is present. Ankuresh Bhattacharya. Okay, guys. Thank you for your time. So I will try to communicate with the leftover participants and see if they can join. So all of you can join that session 2. Now our guest will be there. So some session is going on. After that will over. Our guest, Mr. BB Swine, secretary MSME. He will be joining us. So you can join on hall 2. And meanwhile, we are facing some technical issue due to which the hall 1 is not getting live, particularly the videos. So please be bear with us. Thank you all. Thank you for your time. And by tomorrow, I will let you know regarding these hours from this group. Thank you all.
Okay,
Thank
sir.
you,
Thank
sir.
you.
Okay, thank you, sir.
Thank you.